Mithal Stern. I'm the interim provost and executive vice chancellor and professor of statistics at the University of California, Irvine. And it's my great pleasure to kick off this webinar, Advancing Precision Health. And I'm going to do a brief introduction. The webinar today is an activity of the Precision Health through Artificial Intelligence initiative at University of California, Irvine, which is headed by several people that you hear from today, Leslie Thompson, Peter Chang, Daniel Chow, Xiaohui Xie, and Suzanne Sandmeyer. This is an initiative that was provided seed funding from the Provost's office and has been engaged in a number of activities to flesh out what precision health means for artificial intelligence and do some valuable research in that space. Just earlier today, in fact, I had the good fortune to get an update from the PHAI, the Precision Health Through AI team, on some of the recent work they've been doing analyzing data from the COVID cases in the UCLA Health Center. And it's really quite spectacular work, really just dips your toe in and shows you the potential of modern analytic tools in the space. And so we're very fortunate today to have a really nice program. I heard Professor Geschwind, one of the first people I heard actually talk about kind of precision genomics and things like that. It's hard to know when. Time has lost meaning in the modern COVID world, but he's a wonderful speaker, as are the other speakers on the program. So again, welcome. Thank you so much for being here today. I'm going to turn things over to Leslie Thompson, Donald Brent Professor of Psychiatry and Human Behavior and Neurobiology and Behavior, and one of the leaders of our Precision Health Through AI initiative. So welcome, Leslie. Thank you.
Thanks a lot, Hal. Sorry about the video feed there. It's really a pleasure to have this as our inaugural distinguished lecture series that we had hoped, obviously, to have in person, but it's wonderful that Dr. Geschwind's agreed to do this remotely. And this is a series that we're going to be having several times a year, where we have an external distinguished lecturer and then internal speakers as well paired up with around that topic. Today's topic is precision health overall, and you'll hear several different talks that relate to that. So it's really my, first of all, some logistics. If you have questions, we'll be having questions after each lecture and then a Q&A, and there's a Q&A tab down on the Zoom video conference. Rather than the chat box, don't use the chat box, that's not working, but the Q&A, and those online can use chat, and those questions will be transmitted to us. And then at the end of the series today, we will have a sort of fireside chat Q&A that's general for each of the speakers and discussion session. So it's really a pleasure and honor to introduce Dan Geschwind, who's our distinguished lecturer today. Known Dan for many, many years. He is the Gordon and Virginia McDonald Distinguished Professor in Neurology and Psychiatry and Biobehavioral Sciences. Obtained his MD PhD at Yale and then came to UCLA. Has trained in neurobiology, genetics, clinical neurology, and he founded the neurogenetics program at UCLA in 1997. And for many of us who've known him for a long time, he's really laid much of the foundation for functional genomics using systems approaches to study neurodevelopmental and neurodegenerative diseases. He's identified many of the genes associated with autism spectrum disorder. He's launched all sorts of different scientific resources for the community, for the autism community and others. He's a member of the Institute of Medicine and has received all sorts of different awards. And he also is the director of the Precision Health Center at UCLA, and he'll be telling us a lot about the structural things that he's put in place, the ability to use both clinical information and omics approaches. And so, not to take any more time, welcome, Dr. Geschwind. Thank you so much for being here.
Thanks so much, Leslie. I'm just making sure you guys can hear me, and then I'll find I need to start the slideshow. Start that. Do I need to swap displays, or are we good?
You're good.
So really, no, thank you. Very kind introduction. Of course, the reason, not the reason I wanted to come, give a talk was to interact with all of you and to have a chance to interact with all my colleagues at UCI, which is unfortunate. We don't interact more than we do, given that we're only an hour, 15 minutes away or so. Actually, today, maybe even 40 minutes, depending, because the traffic is so bad. So, but anyway, it's great. And I really do think that the kind of work that we're talking about here, that you're going to hear about today, is best done collaboratively in large groups. And I think the UC health systems, medical systems, along with their other non-health campuses like Berkeley and Santa Cruz, are in a position to lead the world in this area. And certainly our goal is not world domination, but I say that tongue in cheek, but the point is that with all of our patients, with all of our diversity, with all of our expertise, we really have an extraordinary opportunity. So I'm hoping this is the beginning of us starting, to really work even more closely than we have together to achieve precision health in some ways. It will take a village. So my title is a hybrid title. The neuropsychiatry part, I'm not going to get to till maybe even not if I go slowly, but achieving precision health and neuropsychiatry using integrative genomics. Our work is integrative genomics in the lab. But what I've been doing at UCLA since 2016 has been developing an institute that's cross-disciplinary and cross-departmental and cross-school to begin to achieve use the body as a source of big data. And so what I'm going to talk about is, first I'll introduce precision health and how we think about it, a little bit about genetics and why we're doing it. And I'll talk about some of the resources we've been developing and little snippets about what we've been doing with it. And then in the end, I'll kind of hopefully end, it will be a hopeful note about where we're going. So what is precision medicine? I think this is the best. This is the Obama demonstration, delivering the right treatment every time to the right person. This was his administration's statement. Now, of course, that sounds very simple, and it is. But as Niels Bohr said, prediction is very difficult, especially if it's about the future. And to be able to give somebody the right treatment every time to the right person, it's not just about making the right diagnosis in the present. It's about a lot of prediction. And so this is a very difficult area, and it's fraught with a lot of challenges, and you're going to hear more about them today. But again, in my role as an introducer, healthcare tailored to you. That's the promise. Now, the point is, it doesn't mean we're going to have 7 billion different ways of delivering healthcare. It's just that rather than treating everybody with hypertension the same, or everybody with a particular neurologic or psychiatric disease the same, we'll subgroup people to identify key pathways that then we can hopefully target to change outcomes in a positive way. And the idea is you wouldn't wear just any pair of glasses. You don't go and just say, oh, just give me a pair of glasses. You get glasses that are tailored to your vision. And it is pretty amazing that we don't do that for many other things that we do. It's just a paradigm that emphasizes the ways in which disease risks are unique and different among different people, just like other more obvious characteristics, like the way we look. And so it's an approach that integrates genetic, environmental, and other forms of health-related data to develop more personalized diagnostics and treatments closely targeted to individual patients. We use precision. Some people don't like that term. I think, as a physician myself, that we've always done personalized medicine. When I'm seeing a patient, it's that patient that I'm caring for. So we're trying to achieve more precision. And I use the term precision health because it has to do with not just waiting till somebody comes to us for a medicine or treatment, but actually this predictive and preventative part of things that I think has so much promise. So it integrates genomics. Genes can suggest what diseases we might be predisposed to. And of course, the phenotype is the patient and what they might be coming to us for. And then there are other factors like diet, exercise, medications, microbiota, and even where we live that might influence us. Now, you're going to hear me talk a lot about genetics and genomics, and one of the reasons I do that is because genetics and genomics is something that's entirely measurable, it's entirely studyable, it's within the real realm of studying, and is tractable and efficiently looked at. The lifestyle environment factors we still don't know enough about, but there are plenty of them that fit in the same category as genomics and genetics, and that they have a very large effect, a measurable effect, and something that we actually know. And I'll show a little bit of that as well. So the idea is to integrate these data when available and to make them more available and to understand more about them so that we can get a more precise snapshot. And the major goals, as I kind of see it, is, from a practical standpoint, diagnosis, improving diagnosis, prediction, in other words, before somebody's sick, knowing is there something we can do to avoid a disease, diabetes, for example, or predict outcome. When we have a child with autism who's 2 or 3 years old, it can be very, very tough to tell parents what that child's going to look like at 9 or 10, and having better metrics for that with genetics will certainly be helpful. Prevention, as I mentioned, and optimized treatment choice. These are all kind of interrelated in a way. But once we make a diagnosis, maybe we can do a better job treating. And of course, from a research standpoint, this becomes a virtuous cycle in that as we discover things, it will help us understand diseases that we don't understand that well, especially neuropsychiatric disorders, which we don't understand most of their mechanisms. So a first step is actually a kind of link off to the side to basic and clinical translational research to understand disease pathophysiology better, and then feed that back into the healthcare system so we have a true learning healthcare system. So why genetics? It's a measurable cornerstone of individual differences. It's not fully predictive. All human disease consists of an interaction between genes and the environment, period. The DNA sequence of the human genome is about 99.4% identical among individuals. Most of it is the same. The 0.6% difference, though, accounts for all of our individual differences in many aspects: predispositions to diseases or disorders, disease courses, and response to medicines, in addition to things like hair and eye color and things that we appreciate. It's due to both common genetic things, that is, things that are shared by more than, let's say, 5% of the population, called single nucleotide polymorphism, or rare mutations as well. There's an increasing appreciation for the role of rare genetic variation. And then over the last 15 years or so, we've begun to appreciate that it's not just the single base pair mutations, there are actually changes in the copy number of our DNA and in structure of the DNA, and that actually affects almost that 0.5% of the genome, because these cover larger territory. So the average human genome differs from the reference at about 5 or 6 million spots. So there's a lot of space to look at, but we can measure those 5 or 6 million spots themselves, the common things, for, essentially for about $40 or $50 per person now. And so it costs maybe 10 times as much to assess the whole genome, but to look at these spots that are common, about $40 or $50. So this is tractable. It's a definable space. And in addition to individual genetic differences, there are also, of course, environmental and lifestyle differences, and we'd like to be able to measure them. But the whole point is that one size does not really fit all. So the two major drivers, like this kind of knowledge to a first approximation, has been around for a long time. But what's really happened is dramatically decreasing DNA sequencing and massively increasing computer power. And they're very interrelated to each other and have really pushed this. On the left is the DNA sequencing cost. You can see that the Moore's law, which is the integrated circuit, that the density of chips on a circuit is going to half every 18 months, that's shown in green. That's an exponential. What's shown here is in the mid-2000s, that sequencing cost even deviated from that exponential decline. There was a disruptive technology that came in. And so now sequencing our human genome costs about in the range of about $500. Of course, along with that, enormous changes in our ability to capture information and to compute on it, which is essential to actually leveraging all of this genomic and genetic data, as well as the clinical data, as well as capturing eventually environmental data and other health data. So our signature initiatives in our Institute for Precision Health, and it goes a little farther than this, but I'm just going to mention the major 4 signature initiatives. We have a population health initiative that's essentially a biobank connected to the electronic health record, or EHR, where our first blush is to genotype for common genetic variation and sequence up to 150,000 people to start with of our patients. We have a California Center for Rare Diseases that's the umbrella under which our genomic medicine is being done, where we sequence patients, and that's been going on since 2012. And the idea there is it's clinical care for patients with rare diseases, research and innovation. And one thing to realize is that even though rare diseases are individually rare, they account for 7 or 8% of the total disorders that we see in this country. So as a group, they're not rare at all, and they're tractable. We have a Center for Smart Health, which is essentially the electronic and kind of mobile health element, remote monitoring, wearables, mobile tech, and sensors, and precision diagnostics across other areas that are, let's say, more common and not rare. So cancer genomics, a number of translational cores, as well as the Depression Grand Challenge, which is not necessarily, it's separate from our institute, but our institute's infrastructure is helping to support that activity, which is to tackle and treat and cure depression. So the Center for Smart Health, which I'll just show this one slide, is run by Arash Naeem and Alex Bowie, and is really a merger of the computer science department from the School of Engineering and David Geffen School of Medicine. And essentially there we're taking this kind of information on diet, lifestyle, environment, EHR, health center data, and pulling that all together. And SMART stands for systematic, measurable, actionable, resilient, and technology driven. And so the kinds of things that are being done there are projects that are looking carefully at patients who are thought to be at high risk for readmission or high risk of falling and injuring themselves at home, and seeing if we can predict that. And then once we can predict, which we can, does knowing that allow us to intervene and change outcomes? Those types of projects. There are all kinds of other wearables and monitoring, including some very sophisticated patches that can measure dozens of metabolites in parallel, et cetera. The population health project is called Atlas, or ETLA. We were thinking this is essentially creating a powerful and robust cloud-based clinical and multi-omic data resource from at least 150,000 patients. We don't necessarily intend to stop at 150,000, but we're building towards 150,000. We now have over 60,000 patients consented, 35,000 opted in, 22,000 genotyped samples. Actually, it's now 30,000 from this slide. We had to stop early March. The way that we do this, though, one of the innovations is the universal consent, shown here, where we recruit people using a universal consent that's actually an iPad. It's a set of videos that are translated into 8 languages, and it's been deployed in English and Spanish. That's in a number of clinics and places where people get their blood drawn and get tests. It's a 4.5-minute video consent that, because of the IRB reliance, some of the other UC campuses are actually adopting this as well. It's turned out to be a very inexpensive and very effective, not quite passive, but almost passive way of consenting patients, much more effective than just relying on MyChart or the EHR. We have central biobanking. We're doing SNP genotyping with the Illumina Global Screening Array. And of course, it's connected to some high performance computing. just to give you an idea, in the EHR piece of this, the kind of clinical and genetic data integration, we have an Azure cloud-based system that was built by our OHE, our Office of Health Informatics and Analysis. And so it has lab tests, ICD codes, de-identified patient demographics, visit info, medications, et cetera. 5 million. We're also then connecting that to the genotypes right now, which is what we're doing in-house. And we've been looking for partners to do the sequencing with, and that's an ongoing issue because the sequencing is a lot more expensive than the genotyping. And we really do think that the EHR is incredibly valuable on its own, but even more so with genomics and genetics. Not just, we're starting with genotyping, but we hope eventually to have methylation and other data like that on these patients as well. And this is a paper that was driven by the Mount Sinai group, but it's an international consortium of biobanks that we're part of, academic biobanks mostly, and some country biobanks. Ours is in there. You can see it's one of the smaller ones, but we were almost the last starting. But since then, we've doubled in size. So we'd be kind of an average size there, but we're between 20 and 100,000 still in our biobank. Why is it promising? Sample sizes are large, populations are diverse, and there's longitudinal data. But from a standpoint of somebody who's studying neuropsychiatric and brain disorders, there's some other interesting things as well. This is all very true, and I think this makes this kind of research the future. But another thing that we've always known is true is that neuropsychiatric disorders have significant overlapping genetic components. And this is just from a review that I wrote with Jonathan Flint a few years ago, that's just showing as we get more and more cases studied in our various cohorts, shown in the orange is schizophrenia, which when it got over 38,000 at 108 genome-wide significant loci. This was a huge advance. Autism, which is below it, we didn't really have any yet. Now we have 5. But the major point is that genetics is beginning to work as our sample sizes get into the right size. We're finding a lot of genetic loci, both for major depressions, psychosis, autism, et cetera, the ADHD. But the issue is that each of these samples is being collected by highly specialized investigators in certain, with the exception of a few countries. And so my group might be collecting autism cases, but we're not paying attention to any of the other illnesses, not just mental illnesses, not any of the other illnesses. What are what's the family's history of rheumatoid arthritis? We don't know that. And one can see here that the genetic correlation between these disorders is also quite high. Even autism and schizophrenia, it's 0.2. Now, that's telling you that most of the genetic liability is distinct, but they share about 0.2. So the point is that that genetic risk isn't disorder specific. It affects organ systems, which include the brain in certain ways during development and, maturation. And that can give liability to a wide variety of syndromes, and understanding that pleiotropy is one thing that I think having the electronic health record is going to be extremely helpful with. In other words, understanding the relationship between different disorders to each other in ways that we never have been able to do before, with a level of precision. So we think that it can help approximate population level research because it's less biased by the phenotype ascertainment, how I collected that patient. So one of the things is that, that people say is, well, the phenotypes are crap, and it's very difficult to get good information out of these medical records, blah, blah, blah. Well, true. Believe me, as a physician, I know that more than anybody, or as much as anybody. These are 3 graduate students, really talented, in Bogdan Pasanuik's lab, who've been working on this for the last 18 months. And I'm just going to present some of the stuff here, but the point is that you can get meaningful phenotype information, and it's likely going to be better than the kind of phenotype information that you might get out of a population health registry in a country. But again, all of these kinds of data have caveats, but I just want to go through some of it with you. So phenotyping by ICD codes is very, very tough. If you just pull out an ICD code, like how do you decide whether somebody has diabetes, et cetera? And so they're used internationally and across institutions to represent diseases, symptoms, or injuries. So you have a code for autism or a code for ataxia, et cetera. But they're used for kind of billing purposes and kind of large scale checking the boxes. And they can be kind of at some level too detailed and at other levels not detailed enough. Like it might say broken foot on the left side. Well, you don't care whether it was left or right side, you just care about broken bone. So again, one has to kind of aggregate and summarize these kinds of data and use them to get a true diagnosis. And so active diagnosis and history of a disease are in different categories. Diseases can be separated into many categories. And they're not always used to denote a diagnosis, but can be symptoms, place of occurrence, or other descriptive information. And so this is from this paper from Wei et al. And PLOS One that really came up with at least a first approximation solution that we've started to use that's quite interesting, which is called Phi codes. That is, you pull together a whole bunch of different ICD codes, and I'll show you this a little bit, to kind of begin to get a better handle on, so it's aggregates of different ICD codes that give you a better handle on what the phenotype actually is. And this is just showing in our patients, all in red in the background, the different Phi codes that are there. Like if we look at our 5 million patients, 33% have respiratory, about 23% have neurological, and among the neurological, this is how they kind of fall: headache syndromes, chronic pain, migraine, insomnia, et cetera, all the way down. Migraine with aura, it's about 2%, et cetera. So one can use these. And so then the question is, how do we begin to validate them? And by no means am I saying that these are entirely valid, but I'm going to show you that they're a useful starting point. So one of the things we can begin to do is say, if I use the Phi code for like Alzheimer's disease or sickle cell anemia, or develop something for autism or multiple sclerosis, is it pulling out meaningful biology? And so, we can ask questions about Alzheimer's. Is it enriched or depleted in those over 65? Well, yeah, the odds ratio is 52. That's telling me I'm probably pulling something out of, it's probably reasonable. How about sickle cell anemia? Well, it should be in the people who are African American or self-identify as Black in terms of race. And so yes, the odds ratio is 29. Autism, male, it's almost spot on what we find in our clinical symptoms, in our clinical cohorts, 3.8, et cetera. So at least when we look for reality, checks, we're finding them, that what we're finding fits with how these diagnoses would actually work in the population or in our very detailed clinics. Spina bifida, 3.0. That's something that actually wasn't there before. That wasn't known, that it's more common in people of European ancestry who are saying they're not Hispanic or Latino. So again, that's potentially an interesting new finding that we have to look into. But it's just giving you an example of how valuable and interesting this work can be to look at. Another thing one can do with this, though, now that we have genetic variation, is begin to find new associations. So for example, we can take a variant, this IRF4, that's a well-known risk locus for skin cancer, and ask, how's it related to all the other traits that we have if we take them across all organ systems and apply this kind of Phi code? And what we see, this is a genome-wide association study and kind of a Manhattan plot. The y-axis is the negative log p-value. And you can see that, lo and behold, we get skin cancer. It gives us a very, very strong, people who have, GWAS hit. Okay, but other things like squamous cell, basal cell, and even actinic keratosis, which is some of these things are kind of relative to, our markers of sun exposure. And this is telling us that this genetic variant actually predisposes to those as well. So really quite interesting. So again, this is just giving you an example of how one can begin to use these data. And There's so much that one can do. So I hope that I've at least given you a sense that these, and we'll talk about it a little later. I'm gonna get back to it. One could give, Bogdan Pasanuk could give you an entire lecture on Phi codes. And one of the things we've been trying to do is also refine some of these. So we get experts to look through 200 charts and use the Phi codes and see if we can actually add things that make them better, especially for disorders that we want to work on. But we really do believe that they're a really nice starting point for starting to do this kind of EHR research or EHR genomics research as well. So when we get back to the major goals of precision health, one of the main things is diagnosis. And I mentioned that we've been doing a lot of sequencing for diagnosis. So I'm going to talk to you for a second about the California Center for Rare Diseases at UCLA. It's precision diagnostics that started via clinical whole exome sequencing in 2012. And again, this was started by Stan Nelson, who's shown there, and Eric Villain, who's no longer at UCLA, and their colleagues in 2012. And then the Undiagnosed Disease Network started, and they joined that and have received some grants for that. So we've been one of the early founding members of that. And these are just a few articles that show the utility of this, both in families and help guiding care in about 10 or 15% of patients. This is the network now as it now exists. Stan and Christina Palmer and Julian Martinez are the PIs. Nagme Durrani is the lead genetic counselor. There's sites all over the country. They basically try to solve medical mysteries by doing exome sequencing first. This is the kind of diagnostic rate from 2014, which was 2 years into this. I think it was the first 100 patients or so, or several hundred. It was several hundred at this point. Basically a conclusive diagnosis in 26%, a potential diagnosis, which was in 22%. And that means that things don't fit perfectly, but it could be. And then novel gene discovery in about 3%. This is showing the type of mutation. Most of what was done was in children, and most of it was trio sequencing. And so it's most powered to identify de novo mutations. And that's what most of the mutations were. And this was published in JAMA in 2014. And it really showed that, there was this huge diagnostic odyssey in many of these patients. They had seen multiple doctors, experts who had missed the diagnosis because they might have been a little bit atypical or it just wasn't thought of. And the exome sequencing nailed it. And so, this costs a few $1,000s with all of the, there's a, the whole process doesn't just involve the sequencing. There's a clinical genomics data committee, just like a cancer committee that sits and meets every week and that looks through all the cases. It's like a case review. So there's added cost to this, but up to a few years ago, UCLA had done this with 67 hospitals throughout California and locally, and was doing this as a service. Even in neurologic diseases, and this is from one of my colleagues in neurology, Brent Fogel, one can see a very high yield. So this is from 4 different papers, just basically the number of patients or families shown on the left. And basically you can see the rate of neurologic diagnosis is very similar across these. These are in adult patients, mostly with ataxia or other undiagnosed neurologic disorders. So we believe this is a very high yield clinical test. And in some ways, the testing is getting so inexpensive that I believe that in the future, hospital systems will essentially just sequence every patient to have this information because it will save so much money in terms of care in the future. So these rare Mendelian forms are estimated to account for about 7% of patients. At places like UCI, tertiary care centers and UCLA, this is going to be higher, closer to 10%. This is just one of the syndromes that Valerie Arboleta, who was a trainee at the time, who's now an assistant professor in pathology and was an MD Ph.D. This CAT6A syndrome, which was a brand new syndrome. Well, we don't have any treatments for this, but now that the gene has been identified, one can begin to work in the laboratory to do that. In fact, that's what she's done. But what happened is one of the families created this foundation who were one of these patients. And now they've been able to connect using the web all over the world. And it's kind of created a home and hope etc. I think those of you who are clinicians and those of you who might be parents can understand how this kind of thing, knowing what you have, even if there isn't a treatment, is a valuable first step in providing hope for the future. This is a story of many. Here's a girl that had severe swelling of her lymph nodes since infancy and recurrent infections. Years of treatments failed. She was missing weeks of school yearly. Manish Bute's lab discovered that, who's the head of a pediatric infectious disease immunology, discovered that her lymph nodes are packed full of a particular kind of T cell. Whole exome sequencing revealed a new mutation in a gene that overdrives T cell activation in PI3K. There's the mutation. He spent a few years in the lab seeing what was the mechanism. But once he understood the mechanism a little bit, he was able to try a drug that was already on the market that blocks this path, the downstream parts of this pathway. Her disease has been halted, and she's now in the 6th grade because this is a year old doing well. This is just an example. Of course, this doesn't happen every day, but this is where we're going with this precision diagnostics. Can we do better than whole exome sequencing? The answer is yes. We've moved to a pattern where we start with whole exome because it's cheaper. When that's negative, we go to whole genome sequencing. And this was part of our CTSI project that we ran over the last 4 years, where we asked, if we take those people that are negative in the first phase of the undiagnosed disease network, and we asked, what's our yield with whole genome sequencing? And then on top of that, what does RNA sequencing yield? Because whole genome sequencing will often identify potential non-coding mutations that might be pathogenic, but having the RNA could theoretically help. And in this paper that led by Hain Lee, she showed indeed having both of them together helps massively. And so the total molecular diagnosis was 38% instead of 26%. So it added 12%. I should say here, she also hasn't counted those people found with whole genome sequencing to have a mutation in their exome. Sometimes the exome isn't deep enough, it misses it at the first try. So actually another 10% are identified on whole genome sequencing that were just missed by whole exome sequencing, in addition to the added, mostly structural variation that's added by the whole genome sequencing. So now this is our kind of preferred approach. This is the distribution of the patients. About half of them were not, when we say neurology, it could be pediatric neurology, it could be developmental delay, et cetera, or autism. But this is the distribution of the patients. So very high rate of molecular diagnosis, and the cost of this is way less than the tens of thousands of dollars that are spent getting MRIs and years of people searching on the diagnostic odyssey for what they have. So this is an argument for a genetics first approach to patients that are challenging to diagnose. I'm going to give you another side of this as well. You can actually use the electronic health record itself to identify undiagnosed patients. So here's something again from Manish Bute and Bogdan Pasanuik. It's a collaborative project that's been driven by this graduate student, Ruth Johnson. And so Manish, his lab focuses on a lot of these very rare immune deficiencies. And these patients get recurrent ear infections, chronic sinusitis, bronchiectasis, enlarged spleen, enlarged lymph nodes. Now, any of these things on their own are not that specific. And these are very rare. But there is significant delay in diagnosis and treatment. In fact, when we look using our EHR, it's an average of 2 years of delay from the first onset of symptoms. Or, well, it's actually more than 2 years. It's more like sometimes more than 3 years. I'll show you something in the next couple slides. But could you, by taking these symptoms or phenotypes, identify people at high risk for this without without them coming to, prior to diagnosis? So this is just a cartoon showing, these are the, some of the associated features, and you can begin to build a score that, can you identify patients that have more of the symptoms? And you can identify those with a very, very high risk score and just rank every patient in the health record by those scores. In doing this, actually, so you could take, so here's an example. You have 13% of patients have URI, but in the combined immunodeficiency, it's 24%. Chronic sinusitis, well, that's 4%, but in this, it's almost 50%, so increased tenfold. Asthma, common, slightly more common, 4 times more. Bronchiectasis, uncommon, but quite common in this. If you add all those together, it's 4% of patients. Obviously, this is, all of these patients don't have that, but you can rank these people, you can, on a score. And actually, Manish Butte had the top 12 people come in, and all of them had CVID that was undiagnosed. This is an example of a patient that actually had been diagnosed with CVID in 2015. But one can see that if one had just, and on the top are the diagnostic codes entered into the medical record. And if we had just applied this kind of machine learning approach and asked, do the fee codes, what percentile of patients does this put this patient in with regard to these fee codes that are related to CVID? Over a year and a half before they would have been diagnosed, before they were diagnosed, they were tested and then given immunoglobulin. You can see the testing and the diagnosis is shown there in red. So when we actually, when Manish and Bogdan actually looked back, they find that on average, we would have identified the people 2 years before, and that would have saved them enormous morbidity and in some cases, potential mortality. None of the patients, fortunately, had died, but in that case, but there are cases where people do die from this within the first 2 years. So again, one can use this kind of approach to identify high-risk patients and then have some kind of screening capability or call them in, et cetera. That's the idea here. And it's pretty remarkable. So I think what we're learning from some of this, and again, it's very early days, and I've just given a few examples very quickly and very briefly. Genetics first is a clinical reality and should be expanded. EHR signatures leverage common patterns of phenotypes to prioritize patients, even with extremely rare disorders. And these risk scores can be used to prioritize patients in a clinical setting. Now, how about other kinds of risk scores? Here I talked about a phenotype risk score. How about genetics? So if we get to prediction, disease risk, and outcome, we can use the phenotype from the EHR, maybe combined with genetics, it will even be more spectacular. So imagine if we took those patients who were, let's say, the top 100 patients with that, and we had some kind of genetic markers. Could we identify those that are in the top 10% of phenotypes and in the top 10% of genotypes, right? And that would give us the group that are, very, with high sensitivity and specificity. We don't know that yet, but that, but I would predict we'll be able to do that. So we can use genetic scoring called a polygenic risk score. It's kind of a composite score where, we all fall on a normal distribution in something. If you have a baseline risk that's at 0, and you can either be to the right, which is increased risk, and that means you carry many common risk alleles, each with very small effect size. So none of them are predictive with regard to disease. But if you have hundreds of them or thousands, they might be highly predictive. Or those with decreased risk who carry few. So you can identify using this the 5 or 1% of the population at highest risk. And the question is that really useful? So this is a paper from Nature Genetics that basically does this for various forms of cardiovascular disease, doing exactly what I just showed. And it showed that if you identify the people in the top 0.5%, they have a 5-fold increase. Top 2.3%, shown in A, 4-fold increase in risk over those who fall on the mean. And again, if you just took cases of people with defined coronary artery disease, you can see that there's a general bump in their polygenic risk, and it's significantly different. But you can't use the average. You have to use this to identify people in the high risk score. in the highest category. Let's say the top decile, the top 0.5%, 1%, 2%. And those are people whose risk is almost as high as if they had a major mutation. We can see here that to have an odds ratio over 4, which is almost at a Mendelian level of mutation, that was, again, 2.3% of people with coronary artery disease, 1.5 with atrial fibrillation, et cetera. But any 5 of these, together, if you were in the top 5%, your odds ratio was over 4 for one of these diseases. So it's just showing that, in some cases, and this has been done in cancer and breast cancer and colon screening, and I think that this will be combined to identify people at very, very high risk and very, very low risk. Those in the middle, it really won't be able to do much with at this point. And we're moving into this with psychiatric disorders as well. But you could imagine, as we understand more phenotypes, we could identify people who have a better, treatment response, avoid, do pharmacogenomics, and avoid those who are going to have bad treatment outcomes as well. So how about prevention? I can predict and I can find those with 4-fold risk, but can I prevent? I think the answer is yes. And it's not surprising that most of that likely has to do with environmental factors. So here it's showing in 3 different studies, those with favorable lifestyle in blue, intermediate in gray, and unfavorable in red, that's defined by the authors, in low, intermediate, and high pools of genetic risk. So it's environment by gene, standardized 10-year coronary event rate. And one could see that if you're in the lowest group, environment, will really, having an unfavorable lifestyle is really, really bad. But it's really, really bad if you're in the highest group too. But it's double the risk. In other words, if you are in that high group and you high genetic risk and you have a bad lifestyle, your risk is double somebody who has a favorable lifestyle. So even if you're at the highest risk, you can bring yourself very, very close to those at an intermediate or lower risk by modifying diet. But knowing that is extremely important and can help direct treatment. So yes, we can prevent or mitigate against this risk, and it might be helpful to identify those people and work extra hard on getting them to change their environmental factors. But one sees that the environmental factors have an effect throughout the genetic risk pool. One of the problems with these risk scores to date is that they're done in mostly European people, and they don't really apply to non-European cohorts. So we need to do work to do that. But also, diverse race and ethnicity are essential to accurate genetic prediction. So supposedly pathogenic variants, if not evaluated in multiple populations, can result in genetic misdiagnosis. This is a paper from Zach Cohane from a few years ago that basically shows the frequency, the minor allele frequency. This TNNT2, a gene mutation was thought to cause a cardiac disease, but because it was low prevalence in certain, in the white American population. But actually, if they had been looking across diverse populations, they would have seen, no, it's just population stratification. So at least they say that 163 pathogenic variants should be reclassified as benign. Luckily, in our areas, we have one of the most ethnically diverse in the US. In fact, California almost is like a map of the world. In LA County, there's no single ethnic or racial majority. And at UCLA, our patients aren't exactly representative of this. In the county system, they're more skewed, but we're about 35% non-European. So I'm just going to give you an example of something that we took a look at. Again, Bogdan and his group were taking a look at this. So as we're genotyping, so in our Precision Health project, we've been using these global screening arrays, but on them, we put about 9,400, variants that are related to disease. Some of them aren't causal at all. But some of them are like BRCA1 and BRCA2 mutations, and others are just risk variants. But we just put those on to kind of augment the array and just kind of understand how those diseases and that risk might be going in our population. So we took populations that had at least 100 individuals. We didn't include those with unknown race or unknown ethnicity, and and we filtered through these through different self-reported race and ethnicity. And we can also stratify these patients now because we have their genotypes as well. So here are two things that are variants that we found that hadn't been clinically reclassified, but that have a very, very different, but probably should be because they're at high frequency, way higher than the disorder in particular populations. How about optimizing treatment choice? Are we really there yet? So I've talked about using sequencing for diagnosis, prediction using polygenics and other scoring, prevention, even combining genetics with the environment, now optimizing treatment choice. Well, we already have, this is an older paper here, ongoing phase 3. This drug was now approved for DMD. There are a whole bunch of now gene therapies that are approved for specific diseases that can be clinically diagnosed and genetically diagnosed. And some of them are specific, especially in cystic fibrosis, to the exact kind of mutation that's there. And we even have like biomarker-based precision medicine. So in MS versus something that was part of MS called neuromyelitis optica, which involves the eyes and the spinal cord. It's a kind of specific form of MS. Turns out that those patients have an aquaporin 4 antibody, and they respond beautifully to this drug rituximab, which doesn't have as large effect in general MS, but in this particular form has a huge effect, et cetera. So more and more using biomarkers or genetics, we're going to be able to stratify patients into those that should get a particular treatment, A or B or C, et cetera. And that's where we're moving, even in neuroscience. So now in the last couple minutes, I'm just going to finish with precision health and autism, and then I'll be done, in case you're getting nervous about me going over. So circa 2008, this is 12 years ago, the condition not defined, it's defined by behavior and cognition. There were only a handful of genes. We didn't know much about its etiology, little understanding of mechanism. I can't say we're that much farther ahead in mechanism, but we're farther. And in psychiatry and neurology in general, many major pharmaceutical companies were withdrawing from CNS, and they had little interest in autism. And genetics really changed things. In 2012, I mentioned, we had a number, when the exome sequencing started here at UCLA, that was the year that the first bunch of exome findings in autism were published in Nature. And subsequent to that, between 2013 and 2019, more than 200 major effect genes identified. The genetic testing yields in academic centers is about 20%. So we do Fragile X testing, we do clinical microarray if that's negative, we do whole exome sequencing. That yield is close to 20% and sometimes higher in certain phenotypes or in certain centers. So it is clinically indicated to do a full genetic workup in every patient with autism. We have clearer, but not clear mechanistic models. And many advances, there is evidence for convergence, just like there is in cancer, and cross-disorder overlap. And I just cite one of the papers above. And there are major drug development efforts in autism ongoing, because once you identify a target, just like Manish Bhute's target that he identified, if you understand something about it, there might be a drug for it. And also, when we start to get into prediction, I can show you that polygenic risk, even in autism, this is showing the polygenic risk score, and it's showing in the siblings that are blue. You can see that it's moved from to the right. It has some predictive value, just like the cardiac scores, but it's really only going to be useful, let's say, for predicting people at super high risk, for those who are in, let's say, the top 5 or 10%. And the other problem is, again, this is European. I could show you this in other populations, and it would not look nearly as good. So we have a lot of work to do in African American, Latino populations to be able to really use this in a public health way. So again, I mentioned that genetic testing can identify about 20%, clinical microarray, Fragile X, exome sequencing. And this is just showing some of the mutations here that at part a disease like autism or a condition that affects mental health involves a lot of rare disorders. So none of these, you can see these mutations listed on the left, POGZ, TBR1, ADNP. These are incredibly rare disorders. When you combine them up, they account for right now about 4% of autism. If we predict that eventually, as we discover all of them, they'll account for about 15%. So eventually we'll get there, but there's still a lot that's unaccounted for. So there's a lot of gene discovery that needs to happen. So what we've learned is many genes, none account for more than 1% of cases, except for maybe Fragile X, highly additive effects, and strong pleiotropy. And that none of these genes so far are specific for autism. They can cause intellectual disability. Some of them can even cause psychosis. So there is kind of overlap again. But now knowing these genes, we know how complex it is. We're beginning to understand the overlap. That gives us a way to begin to peel apart the mechanism. But it raises some challenge to the classic genetics paradigm. There's genetic heterogeneity. Genes don't act in isolation. They're components of signaling pathways. And in the brain, they're parts of neural networks. And so one of the questions has been, is there convergence? How do we take the polygenic risk from hundreds or thousands of risk factors and put them together? Can we identify patterns? Do they converge on certain cells or time points? Can we use these patterns to inform treatment development? And so we take this systems biology approach to integrate big data. And so one of the first things we did is begin to ask, do autism genes, these hundreds that we're beginning to identify, do they converge during brain development? And this is just showing a map of brain development. The question is, are they spread all over the place? Do they converge? And we showed that they occur, that most of the large effect autism risk genes are affecting transcriptional regulation very early in brain development. And then later, the lower effect, common variation, affects synaptic development. And we could also show that it affects very specific anatomical circuits that affect cortical connectivity, that is, mostly superficial layers. That doesn't mean that no genes disrupt layer 5 and 6. No, some of them do. But if we ask where they're enriched in these regions that connect brain regions to others. So this is just an example of how beginning to get, we're starting to get a very complex genetic picture, but can we simplify it by asking when and where these genes are expressed, what cell types they're expressed in, what circuits, and are they expressed in certain pathways? And again, to take this even further, this was 2013. In 2018, we began to use the transcriptome or the RNA from brains to begin to understand how the genetic overlap plays itself out in terms of gene expression in the brain, and begin to really have it's a very complex picture, but a very clear and robust picture of what kinds of cells and what kinds of genes and pathways are disrupted in autism, schizophrenia, and bipolar, and how they overlap and don't overlap. And kind of maybe most relevant for the talks that are going to follow, with Mark Gerstein's and Jim Knowles, we were able to kind of create an integrated model using deep learning. And here, Jonathan Worrell in Mark Gerstein's lab created this DSPN, which is this deep Boltzmann phenotype network. And we showed that had really, by looking at these latent, unmeasured, aspects to the data, we combined transcription factor binding, chromatin structure, both attack seq and high C, the largest expression quantitative trait locus resource with nearly 16,000 brain eQTLs. So, with all this enhancer and promoter data and all the tissue across disorders, we're really able to get a much more refined view and also identify new genetic risk loci. So I think that this notion of, relying on kind of one layer of data and being satisfied with that, just gene to phenotype, is gone. We're finished with that. We're no longer going to be doing GWAS and just finding here are the loci. What we really need to be doing is integrating the GWAS data, the quantitative trait locus data, the gene regulatory and transcriptional regulatory network data into models that can predict how the genes are, what genes are affecting these phenotypes and how they're doing it. So in terms of food for thought, sequence early is an efficient paradigm for modern medicine and should be more broadly adopted. Computational approaches can advance clinical care even in rare diseases. The example was accelerating earlier diagnosis. Genetic and genomic data have potential to transform all aspects of clinical care in autism and other neurologic and psychiatric disorders. I showed you that whole genome and genome sequencing yield many new candidates. It's a useful clinical test, and that systems biology in the last few minutes, obviously I gave it short shrift because I wanted to just talk about the other stuff we're doing as a health system, but the systems biology and gene networks provide a framework for understanding convergent pathways and disease mechanisms. And I should say that this computational approach, and I realize I didn't mention it, for the immune deficiency, there's obviously a treatment for that. It's immune globulin. And so those patients can be fully treated. So the earlier you get to them, the better. And that's true for many of the diseases, even rare diseases today. So a lot of people to thank. That's the lab on the right. I'm just going to thank the precision health related people, the people on the left in my lab, many of whom who are starred have now left and started their own labs or in further training. But Claire Lejean-Cher is the deputy director. It says DD, she's the deputy director of the Precision Health Institute at UCLA. But really critical, essential people have been Arash Naeem and Sarah Dry in the Biobank, and Chris Denny, Mike Pfeffer, Eliezer in Iran in developing the EHR de-identified data mart along with the UCLA Office of Health Informatics Analytics. Mike Pfeffer is the Chief Information Officer at UC Health, and his group has been extraordinary. Of course, I mentioned work from Bogdan Pasanuich, Manish Butte, the rare disease work that's been going on as well. I list my lab funding below, but all the precision health stuff has really been funded through the chancellor at UCLA, the School of Medicine, and the health system so far, as well as a grant to the CTSA. So thank you very much for your attention.
Thanks so much, Dan. That was fantastic and really exciting. It was fun to see Vivek's picture there, who's now at UCI from your group too.
Yes,
I'll read a
played a
couple.
major role. Yeah, in the autism work.
Yeah, it's fun having him. So I'll read we have a little bit of time for some questions, and then we'll take some of them during the fireside chat later. But maybe a couple now. One is, you mentioned that, and I'm just reading.
Oh, yeah, no, I can see that. Should I go to the Q&A?
So you mentioned that whole exome sequencing misses some mutations. Is there a way to improve that methodology, or would it be better to go straight to whole genome sequencing and RNA-seq? Talking about better bang for the buck?
Yeah, no, I think like right now the whole exome sequencing costs $280, and we can do whole genomes in the $500 to $600 range when we get a good discount. The issue is that there's also a higher analysis load on the whole genomes, but we're moving, for difficult clinical cases, especially given this, that most of the money actually isn't in the actual genetics, it's in getting 20 people to sit around a table, it's in the clinical case conferences and all of that. That actually has a huge cost in terms of time for the several hours a week, et cetera, that people are doing that. I do think that, I'd recommend whole genome and sequencing RNA-seq, but also, insurance companies aren't reimbursing that, and they may really require you to do this in a stepwise way. But it's probably more efficient to do whole genome and RNA-seq.
And actually, building on that, Suzanne had asked about why maybe insurance companies are so slow to support whole genome sequencing, and maybe the impact of the UK Genome Project, All of Us, Million Vet Project.
Yeah, I mean, it's interesting. Right before COVID, we started to have conversations with some of the very large insurers, and there are a few that are very forward-thinking and very innovative. And and who have sophisticated AI and sophisticated thoughts about genomics. And we've been talking with them about doing some proof of principle projects that could actually really show, use our expert system to diagnose people and then really understand the cost savings so that they could make a real strong case to the kind of shareholders and upper management. But I think that will move forward. In fact,
Okay.
I would love to see hospitals eventually, just sequencing all their patients, because now they're going to identify everybody with cancer predisposition syndromes and other things, some of which can really be prevented.
Right. Can you read that first one? It's it's about the comparison of UCLA and other systems.
Yeah. So this is from Peter Donovan. Actually, in 2016 and 17, I went to Denmark and spent a sabbatical there, and part of that was to work within that system. And they have a number called the CPR number that you can use to connect everything. I'm talking about taxes, every place they've lived, every car bill they've had to pay, credit card, their bank accounts, and their health record. Now, it's not readily available. It's locked down extraordinarily well. It's not even connected to the internet in any way. But Statistics Denmark has created a place where you can go and where some of these things, where there are various registries tied together. And one of those is called iPsych, where they've genotyped everybody with a psychiatric disease within within a certain time frame. And so I've spent some time working with them there. One of the difficulties is they can't actually go back to the patients because they weren't consented. It was done with blood spots. And so one is limited to what's available in whatever registry you're in. And you don't actually have the details of the medical record, which would really be amazing to attach to it, although they're beginning to work on that. But you can't bring people back. But it's still an amazing system. Africa. Our main autism project now is in African Americans, to kind of fill the gaps that I mentioned. And we'd love to be able to do that in Latino population as well here, but it's just a funding issue. But there are a number of projects that have just started in Africa over the last 5 years, especially in psychiatric disorders. And I work with several of these people in South Africa, and then I have a colleague in Mali that I've been trying to help them get things going. But you can imagine how tough it is infrastructure-wise there, especially you're going to diagnose people, and then in many cases you have absolutely nothing that you can do for them. So there are all kinds of other issues. How do you want to be spending your money, et cetera? But I do think it's super important, to be moving into, again, we want to use this. I think we can use this information to avoid healthcare disparities, but there's a risk that it will accentuate them if we're not careful in the way we go about this.
Right. And Virginia had a couple comments about this being challenging with the
and so we have, we can see how this is not a political statement, it's just a fact statement that you can see all, our healthcare system is like Swiss cheese with regard to just COVID or things like that. Well, that's because that is visible, but there's so many other things like these genetic diagnoses that are kind of invisible that we're missing. Yeah. And so you look at countries like England, where they are now, the National Health Service in England, right, is sequencing whole genome, every admitted pediatric patient. Every pediatric patient is admitted to the hospital, it's sick enough to be admitted, gets a whole genome sequence. Wow. And because they recognize that that's a small part of the cost of care and will eliminate tons of other useless testing. And so, but that only works when the provider is the insurer. So I bet Kaiser will move there. And it is really a strong argument. Seeing these pandemics, seeing the hope of genetic medicine and all these, and prevention and prediction works best when when the payer and provider are the same and it's all integrated, so that you can make, when the incentives aren't skewed towards, well, the less I pay, the less I pay right now, right? Because this person's going to move off my healthcare system in 5 years, so I don't care if they have a chronic disease.
All right. So I think maybe what we'll do with there's a question about how the UCs can work together, but maybe we'll save that for the fireside chat because we can engage all the speakers then in that discussion. So Peter Chang, you want to introduce our next speaker?
Absolutely. I think my video is lagging here. Anyways, thank you very much, Dan, for that great overview. It's my pleasure to introduce our next speaker, one of our very own, Xiaohui Xie. Xiaohui's been with us at UCI since 2007. In the past 13 years, he's emerged as a true leader in the field of deep learning applied to the life sciences and healthcare. And given the fantastic overview we just heard from Dan, I think his talk will be a great segue, highlighting one very specific technology. Of course, there are many ways to try to tackle this problem, but certainly deep learning artificial intelligence has a lot of potential and a lot of promise in this field, and in many ways fits nicely with some of these precision health initiatives. And so without further ado, I want to welcome Xiaohui. Thank you very much, Xiaohui, for spending your time with us this afternoon.
All right, can you see my slide? It's good. All right, thank you, Peter, for the introduction, and it's a great honor to be here today, especially to share the kind of virtual podium with our distinguished guest, Dr. Geschwind, and to talk about the AI and talk about the precision health. So next, I'm going to talk about some of the work my group have been doing on applying deep learning to address some of the problems in biology, especially in medicine. So that
can you swap displays? You're currently on presenter view.
You cannot see it? Okay, let me see. Is this okay?
Perfect. Thank you.
Sorry for the glitch. So what I'm going to do here is this. So as you well know that in the past few years, the tremendous progress have been made in the field of the artificial intelligence. So a lot of product have emerged from research labs and become part of the daily life. And some of them you encounter in your daily life, including some of the application I show up in this slides here. So ranging from movie recommendation to personal assistant, to computer games, to kind of smart Photoshop's applications, to more sophisticated applications such as autonomous driving. So one of the key component underlying many of these applications is this ideas of this so-called deep learning. So deep learning is one part of this, one subfield of the machine learning. And then it utilized some of the algorithm originally was inspired by what's happening in the brain. So what we know in the brain is that information processed through the brain in a layer by layered fashion in a hierarchical way. And those features we are able to extract more and more sophisticated features as we go through the different layers of the brain areas for that. So the computer science, it will take some of the motivations from this type of biological information processing, develop models based on neural nets, later on generalize that to include making it more deeper and more sophisticated. And nowadays, many of these AI algorithms, especially successful ones, are built based on these deep learning algorithms. So this shows one of the applications in deep learning. You have input here, and those information are processed in a layer by layer fashion in a hierarchical way. And people hope that we're going to use those layers of neurons to extract more sophisticated features. And those features will be very informative for us to solve challenging problems. So of course, I mentioned that the AI application is not only limited to a consumer product, and a lot of researchers around the world are thinking about how to utilize this recent development in deep learning to address problems in biology or medicine. So of course, many of these problems are more sophisticated than relatively simple problem that people solve in the consumer product related fields level. And for instance, this highlighted that in this interest here, not only in the academic institutions, but also also a lot of researchers from industry are interested in solving these type of problems as well. So today, I'm going to talk about some of the research in my group that we have been doing in trying to tackle some of these problems. So of course, in the biology and medicine, there are lots of different types of data that people have think about applying the AI or deep learning algorithms. So they are ranging from the genomics or genetics data Dan has been talking about, and to phenotypical or clinical data. And so in this talk here, however, I'm going to focus primarily on the imaging type of data. And in this particular modality, I'm thinking about how do we develop algorithms to solve problems that in this medical imaging domains of that. So of course, medical imaging is nowadays has become a essential component in the medical diagnosis. And so, however, there are many different type modalities of images here, ranging from CT, MRI, and PET imaging, ultrasound, X-ray, etc. So include a quite wide range of the imaging modalities of that. So right now, most of the analysis in the clinical applications are mostly done manually. And however, there is a huge interest to automate the medical image analysis pipelines, such that hopefully to make those analyses automated. And our goal is to make the analysis hopefully faster and probably more accurate and more precise, provide more quantitative measurement from the image side of that. So how big is the medical imaging field? So right now, so there are 2 trillion images are done every year globally, and with half of those images are done within the US. So that many people believe that those images are going to be increasingly to be utilized, and it's estimated that those numbers doubles every 5 years. So there's a tremendous amount of data that are already being generated, and probably will be more data will be generated in the future. So the question is, how do we develop a better methodology to handle and to analyze these type of data sets? So there are some issues, the problems that exist in the medical imaging analysis field of that. So of course, in our panel here, we have a radiologist. So hopefully we're going to hear more feedback from them and later part of this seminar, later part of that. So but there are some issues here that pretty well known. So I just mentioned that there's increasing volumes of the images will come up and in the future. And however, on the other hand, we have a relatively shortage of radiologists. So also in general, many of these interpretations of medical images are subject to personal and individual bias. Also we usually do not have a very quantitative kind of measurement on many of these reports generated by the radiologist side of that. So many of these issues here, many people believe that can be aided by developing algorithm AI to address some of these issues of that. So that is also our motivation in working on these domains of that. So in this specific field of the medical imaging analysis, so in terms of deep learning applications, there are many different types of specific applications. So I kind of highlighted some of the applications here. So including the segmentation and localization and quantifications of lesions, and goes to medical aided diagnosis. So in this talk here, I will primarily focus on the segmentation part of that. And if I have time, I will talk about some other applications, but most primarily focus on segmentation. So segmentation is a very important component of the medical image analysis here. So in this CT scans of these regions, what we want and like to know here, so this is raw images that usually see by radiologists here. And for a lay person like me, I can rarely recognize what are the organs appear within these regions of that. And however, it would be nice to be able to kind of recognize individual organs within the regions here. And hopefully later on, we can capture 3-dimensional shapes of these organs. And by doing that, we hope to identify the regions of interest. I isolate different organs from these individual CT scans of that. And hopefully to help to assist the radiologist to make a diagnosis. And also might be useful for later on for abnormality detection. It might be detect regions here, maybe bigger, or the growth of the regions in certain part of the scans of that. And if we are able to segment those different regions, we will be able to provide a quantitative measurement in terms of how big is this region, what is the volume, what is the shape, what are the changes on those regions. And so that will be very useful in terms of measurement of the lesions or any abnormal growth or monitoring progressions of the disease. So that is the main kind of problem we're trying to solve in my group. So however, medical images application is actually not, is very hard problem here. So as you see from this part of the CT scans, you can see those images here, but there are a lot of regions, organs that are imaged through this, in this piece of pictures here. And for trained specialists, they will be able to recognize the organs associated with each part of the images here. But this will require years of training, and for a layperson like me, it will be very hard to recognize them here. So our motivation is, can we develop a system and such that giving an image, a raw image as the input here, such that we can automatically delineate in the boundary of each organs here and highlight each different organ, such that use this input here, hopefully to assist later on diagnosis or detection of disease. And how do we do that? So what do we do here is this. So many of this, fortunately, for many of the images here, lots of data has been generated over the years with detailed segmentation and detailed other labeling annotations. And what we have been doing over the past few years is to develop this deep learning system to train a model that can learn to delineate those regions based on experts' annotations. So one example is showing up in this case here. So this shows you demos of this piece of CT scans in the brain regions that has been delineated through the deep learning algorithm here. And what we are able to do is this, we'll be able to highlight it in colors or different regions of the brain, and you see from this video slides. So with this regions here, with the model trying to delineate different brain regions here, so now we are able to, given CT scans here, we'll be able to automatically show up the different regions of the brain regions in this particular applications. Highlighted potential organs in this case. So far we have done analysis based on relatively, first of all, in order to carry out this analysis here, what do we need to do? We need to generate a large amount of training data set to train our models. So we are fortunate enough to be able to collaborate with a few institutions that they are help us to annotate those data here by carefully delineate those different regions of that. Afterwards, we are able to train our models to be able to segment the different regions from the CT scans of that. So right now, we are able to segment this primarily from CT scans, segment over 60 different normal organs from the CT scan data. Some of them are highlighted in those pictures here. And this picture primarily focuses on the segmentation of the brain regions, but our recent work has been extended from the brain to other regions, to the chest, to the abdominal, and to the pelvis region there. So our goal is here is try to hopefully to provide a more complete, a comprehensive system, such that we'll be able to kind of highlight each individual CT scans, point out the regions, and detect any potential abnormal regions within CT scans, do this automatically. So after those each individual scan has been kind of processed, delineated, and what we can do next is to build three-dimensional structures of each individual organs, and I can visualize them in a three-dimensional way. So by doing that, what we can do is recapture the relationship between the different organs here, and if there's any abnormal growth or the shrink or the growth of regions, and we should be able to detect them using this methodology. So everything is done here in automated fashions. So our model is able to receive the raw CT scans as input here, and then be able to generate those fully three-dimensional recapture the different organs and the relationship between them here. So that's segmentation here, of course, is not limited to delineate those normal organs or normal regions here. They can be used for detect lesions as well. And for many of these applications, I'm going to highlight in the next here is this, so we can apply them for more detect lesions here. So I should point out that, so one of the applications on this organ segmentation is not just for kind of point out the boundary of the regions here, but there is actually a specific field in this radiation therapy domains of that. It's actually required to delineate those regions as a part of the radiation therapy design and planning stage of that. So this is that our work was originally actually developed for that purpose. And last year we published a paper to report this result and demonstrate that those organs delineated through the deep learning algorithm here, in many cases here, we can achieve better accuracy than those experienced radiation oncologists. It shows you, those applications, potential applications, even in clinical domains of that. So in particular, one of the key challenges in delineating those organs will take quite a bit amount of time to delineate those regions by expert. And usually to delineate the entire brain regions, the scans of the 20 organs would take 30 minutes or even more than 30 minutes, a few hours to delineate very carefully. However, with these trained models, we are able to do that within a few seconds of that. So time, it is not an issue with this AI-assisted algorithm there. And also we have to demonstrate that the algorithm in many cases here can achieve better accuracy than those manual annotations. So using the segmentation here, we can also kind of detect and quantitatively measure these lesions and abnormal growth. This will be another applications of the medical imaging segmentation. So I kind of point out a few applications that we have done in the past couple of years. So this one here is this segment those nodules within the chest CT. So nodules will be associated with many potential the disease associated with this lung disease. And I will talk about one of the applications in the later part of that. So in this case here, what we want to know is detect the so-called nodules here. And once we detect them, we will be able to segment them here. And this shows you one of the segmentation returned by the algorithm. As you can see that we can do relatively very well and return similar profile as this kind of ground truth annotation done by the experts there. So once we have the segmentation here, we can carry out this model throughout all the slides through the scans here. And then just like what we did previously, we can capture three-dimensional volume, three-dimensional shape of them. And by doing that, we will be able to calculate the volume of these nodules here. And in many cases here, this volume here might decrease or increase here. And that provides a way for the monitoring on the progression of the disease. So in collaborations with UCLA, our colleagues here, we have done another studies here. In this case here, was trying to detect those MS lesions from the MRI scans of that. So the left side would be raw MRI scans here. You can see there are bright spots here. And we're able to develop a deep learning-based algorithm to automatically detect and segment those lesions. By doing that, we'll be able to automatically report how many lesions a patient has, and this will be done, can be done automatically. And this shows you another application. This application is on this mass detection on the mammograms analysis. This shows you different types of mammograms here. And where in this application, we train deep learning models to detect those mass regions in the show up in the mammograms here. And later on, we're able to kind of not only detect them, but also segment those mass and calculate what are the areas associated with these regions here. Is there any progression on this area when patients take those mammogram multiple times? So this is the part here I talk about what do we have done in developing those deep learning models to segment those different regions, either the normal organs or the lesions associated with disease. So next, I'm going to focus on one particular applications. So that will be related to the chest CT analysis. And the reason I talk about that because, of course, in the context of the COVID-19, there's some interest of look carefully into the chest CT scans of the COVID-19 patients. So I will talk about that. So first I will talk about actually another application. We have in the previous year, before the era of COVID-19, we have been doing some research and in terms of how do we analyze chest CT such that we can do early detection of the lung cancer. So a number of studies has reported that using those CT scans, if we can detect those lesions early, and we can help to actually reduce the lung cancer deaths by significant amount. So one report saying that 30% of lung cancer deaths can be reduced with early detection. So the question, how do we detect those early signs of lung cancer? So one of the hallmarks of lung cancer is associated with something called pulmonary nodules that can show up in the low dose CT scans of that. So this shows you one example of the so-called pulmonary nodules. They show up this as a bright fuzzy spot in this CT scans of that. And however, detect those nodules early is challenging because many times those nodules are very small, can be less than 3 millimeters. To detect those nodules here, and it's challenging because it's confounded with many other factors here, and such as those white bright spots showing up the CT scans of that. And those spots are usually vessels or some other normal organs of that. So the question, the key challenge for the models is to be able to distinguish those nodules from the other confounding factors there. So the way we did this is trained by developing deep convolutional models by utilizing three-dimensional features. And we use the expert annotated CT scans as a teacher signals to tell the models to see which part of the bright spot might be associated with the nodules, which one is not associated with the nodules. By doing that, we're able to train a model from end-to-end fashions and use it to detect those pulmonary nodules. So this showed up some of the result we have, this model result we have done. So this what they have done is once we have the models here, we can build an interface. So in this interface here, if you provide a CT scans here, and the model will be able to go over all the scans of the CT images, and then automatically report the potential nodules appeared within this image there. So this show up in the left side, left panels here. So in this regions here, and then we are able to say, what's the location of the potential of this little nodules here, and what are the, and then we can provide a quantitative measurement because, as I showed previously, because we can segment those nodules and we can calculate what is the area, diameter, or even volume of this individual nodules. So we are hoping that by providing such a system, and then we can reduce the, we help radiologists so they don't need to go through all the scans of the CT, chest CT scans, and then be able to kind of focus on those relatively smaller number of regions, and then take a look at them to see if there is potential issues there. And we'll provide an interface such that if they want to look in the individual ones, they can look more detailedly through multiple views and verify if this is indeed a potential nodules. So, how accurate is such a system? And because, as I mentioned in the very beginning, detecting those pulmonary nodules is a challenge because they're relatively small here. And so we have done some benchmark experiment to compare, to evaluate the accuracy of such models. So this shows the result on a public data set called LIDC data sets. So in this case here, we plot the sensitivity of the model as a function of the false positive rate. So in general, what we observe is this: on this large scale data set that contains over 1,000 cases, we are able to achieve over 96% accuracy in terms of detecting those nodules if we have certain amount of false positive rate. And so if we keep reduce the number of false positive rate, and then we can force the sensitivity reduce, but the false positive rate reduce as well. So in general, what do we say in this? If we are able to report, let's say, 8 or 5 nodules on this left panels here, and then we can achieve 95% accuracy in terms of detecting those small nodules. Of course, once the nodule is detected here, the next challenge is to decide to classify those nodules here and to see which one is social, what are the types of the nodules. And we highlighted some of these guys here. And then the other part is this, which one is nodule is malignant, which one is benign. And then we have built this classifier to further kind of stratify those nodules and classify them, and which one is potentially malignant and will need further follow-up. So then next I'm going to talk about the second application, and in the context of this COVID-19 here, and then we'll think about how can we utilize those methodology we developed previous year for analyzing this COVID-19 CT scans of that. So of course, CT scans of is not recommended for all the COVID-19 patients, but it does recommend for COVID-19 patients with severe symptoms of that. And what we are hoping is this, we can use deep learning here to extract those image features from the CT scans, and hopefully later on that can be useful for patient stratification, maybe for disease progression monitoring, and maybe study treatment effect of that. And as it's well known that a lot of reports have been done to indicate that there are quite a bit of image features associated with COVID-19 patients. In particular, in the report, there are a few high features I highlighted here. So the two most important one is something called ground glass opacity, and the second one is this consolidation. Those are the more or less become the very important features associated with COVID-19 patients. So this show up one example of the COVID-19 patients here from a published work. And what I highlighted in this is the chest CT scan I just showed up in the previous year. This highlighted regions of the patient here. It seems to show up in this bright spots here. And these are called the GGO, the ground glass opacity. And later on for this patient here, after 2 days, you can see that those regions, the lesions here actually become larger and the progress become bigger here. So we think that that might be helpful to be able to kind of monitor the disease by measuring to first of all detect those lesions, and later on to monitoring the size and whether they are progression, whether they become bigger or smaller in the future. So to address these questions here, what we need to do first is to be able to kind of detect those lesions here and then segment them. And fortunately, we're able to have from collaborators, we're able to use quite a bit of annotated data sets here. We are able to train a model based on 1,300 slices, and then we test those segmentation results based on 300 slices. So right now we are able to segment a few types of lesions. And first of all, we can segment the entire lungs. And then later on, we are able to segment the so-called GGO. This is one of the most important features associated with the COVID-19 patient. And next will be the severe part of that is progression from GGO to consolidation and a few other features associated with that. So this shows some of the results from the predicted by the models here. So left side here is this raw CT scans here. So this is from the two patients. And you can see that this patient here have some lesions associated with this pneumonia here. And this is the lesions predicted by the models here. Right now, we are able to generate segmentation. In this scan, we detect there's this region associated with the GGO features. And the second feature is associated with this consolidation part of that. And unfortunately for this data set here, we do have expert annotations, and we can compare them. And we see very nicely there's quite a bit of consistency between this model prediction and these expert annotations. So this shows another example, another patient cases. So in this case here, as you clearly see, this patient is have more severe symptoms in terms of the CT scans of that. And you can see from this part here, this GGO proportion is significantly bigger for this patient. Almost half of the lung regions has been infected with that. And this patient here is also have a significant GGO portion of that. It's slightly smaller than the one in the above part of that. So once we are able to segment those lesions, and then what next we can do, we can provide a quantitative measurement. So quantitative measurement right now, we are thinking about in terms of fractions of the lung regions here, they have this GGOs or have this consolidation part of that. And we have a few other additional features. By combining them, we can provide a little bit kind of image type feature descriptions of this COVID-19 patients that can be done fully automated fashion. So this shows you one actually real example from our local hospitals here. Shows the CT scans of one COVID-19 patient from UCLA hospital. As the left side here is raw CT scan, the right-hand side is this processed CT scan through the segmentation algorithm. So what we can do here, you can see, is that we can segment these lung regions of that and then detect lesions, highlight the type of lesions associated with the CT scans of that. So what we can do here is we can provide a score to measure the severity of the disease here. And by measuring the fractions of the lung regions here, they have this GGO features and which fractions of regions have the consolidation. By doing that, we can provide somewhat a more quantitative measurement in terms of the severity of the patient here in terms of the CT scans. Of course, you can see from this patient here, and it doesn't look very good here because a lot of lesions happens within lung regions of that, and a pretty large fraction. And then scores show up in this side here. For instance, show up the lesions here now represent almost 40% of the entire lung regions. All right, so once we have this quantitative measurement here, and then what we can do, what we did next here is try to characterize how is there an association between those GGO scores and the fractions of the lung regions have those type of GGO image type. And is there any association between the GGO scores and the severity of the disease? And that's what show up in this plot here. And then we have a data set, and then with patient grouped into 3 categories. So left side here is this mild category, and the middle part is moderate, and the right-hand side here is some in the more severe cases of that. Essentially, those are the patients that they are entering the ICU. And what we see from here is this show up the distributions of the GGO scores in each category. What you can see here, you can clearly see that even using this simple GGO scores here, you can roughly separate those mild cases from the severe cases here. And so we thought that would be interesting in terms of the stratifications of the patient and follow up the patient through those CT scans. All right, so I'm going to wrap up the talk here with a final one more part of that. This will be essentially related to how to use computer visions for not only for medical images here, but for general kind of image application, but related to kind of medical application of that. So this shows up one of the applications we have done related to detecting movement disorders. And what we have shown, so this show up here is this patient here. We have this camera in front of the patient here. What we have done here is going to kind of annotate those different key points with this patient here associated with the hand key point and the whole body part of that. By doing that, what we can see is this, once we highlight those key points, we can see that those patients, there's movement features associated with this patient here. In particular, what you see that the hand is tremoring associated with the patient. Of course, what many of you recognize, this is more or less the feature associated with the Parkinson's disease. And we are trying to develop this on a fully automated system such that we can kind of fully quantify those movement here in an automated fashions. And because we can detect those key points here, and then we can plot the movement of each individual key points here. And I'm going to follow up the movement. If the patient is show up the tremor patterns here, or your rigidity movement here, they will show up in those type of curve time series of the curves. So by developing a score, by combining those different movement here, we are able to calculate a score associated with this so-called Parkinson's disease, but completely from kind of computer vision type of features of that. And then we have been comparing those kind of AI system-based scoring system with the experts of that. And then what we see is that there's large consistency between the scores and the calculated systems and those experts. Of course, the system here is kind of pretty agnostic. They are more or less kind of in an automated fashions. All right, so I think I'm behind the schedule. I run a little bit out of time here. What I'm going to just summarize what I want to say regarding this medical image analysis here. I think what I talked about some of the applications my group has been working on in the past few years in terms of developing those deep learning models to analyze the medical images there. However, I should mention that there are quite a bit, some challenges here in terms of medical image domains. Of course, there are many other groups are working on these problems there. So the problem is that the data dimension is usually high in the field of the medical images. So in the case of the mammogram, if we have this 3,000 by 3,000 pixels of that, in general for the CT scan and the MRI scans, that typically we have this volumetric data set instead of typical single images of that. And another big issue associated with this domains is the annotation is usually very costly. In many cases, very noisy. And because it requires expert to annotate those data sets. For that reasons here, and because in order to train those deep learning models, we need to large amount of high quality data set. And in this domains here, it's kind of very hard to generate this amount of data sets here. So unlike in general, many of the machine learning community, many of the data associated with the medical images here, they're not available, publicly available. And as a result, that many of the published results, it's impossible to reproduce this result because of lack of the transparency. So in somewhat, this is significantly hindered the progress of this field. So summary. So what I just kind of wrap up in this is the AI has been already make a significant impact to the health applications. In this talk here, I highlight those applications related to medical imaging applications. I believe they're going to further kind of enroll in this side of that, significantly make progress in the future. However, we need to address a lot of challenges in this field related to data annotation, sharing, and mass transparency of that. But I'm kind of optimistic that the field here is already see some benefit of applying those deep learning models to fully automate some of this analysis of that. And in the future, we might be able to see more and more applications coming from this side. All right, so I'm going to end by acknowledging people involved in these studies here, mostly the students in my group here, and then the funding and the collaborators from various groups at UCLA and the cross campus of that. All right, so I want to thank you for your attention and appreciate your time.
So I'm Suzanne Sandmeyer. It's really a pleasure to introduce the next speaker. In a way, Dan Chow wraps up a lot of what we've heard today and what we'll hear going forward. So Dan received his MD degree at UCLA. He went from there for a residency at Columbia University in radiology, and from there did a fellowship at UCSF. He was recruited to UCI in 2016, so he's a junior, I guess, redundant assistant professor here at UCI. And Dan is one of those rare individuals who really brings together the different cultures that are represented that we've heard from today in computer science and in medicine. Plus he has a huge amount of energy to bridge that cultural gap. So it's really been terrific to have him here on our faculty. He and Peter, who was also at UCSF, have made a real team that has bridged the gap. And finally, Dan is a good ending speaker because he has pivoted quickly. He and Peter have pivoted quickly and applied some of their talents in artificial intelligence to better understanding how to diagnose COVID-19. And so without further ado, Dan, take it away.
Thank you for that introduction, Dr. Sandmeyer. So I want to briefly discuss, how kind of the topics that have been discussed today, and that really is the mission of the Precision Health AI initiative. It's really to improve health and well-being by integrating all of these complex data streams and technology. And so specifically, I'm going to briefly talk about how this initiative has pivoted to identify vulnerable populations, looking at demographics at risk, and ultimately those who may have poor outcomes that necessitate critical care. As Suzanne and Dan and Xiaohui have indicated, precision health really is a multidisciplinary effort, and the same was for this initiative. As you can see, because we're trying to obtain a holistic view of our patients, this really does cross multiple disciplines and experts across the UCI campus. One of the things that we wanted to look at were demographics and populations at risk. And I think it's of no surprise that we have been seeing in media now that certain ethnic groups are being disproportionately affected. These are numbers from UCI. You can see that from weeks 1 through 6, those who identify as Hispanic Latinx have definitely increased. And we ran the numbers last night, and this is now where we stand in week 11, and that trend has kind of gone unabated. But one of the things that we wanted to do was dive a little bit further and see what are the economic and social trends that may be contributing to this. And as we can see that, this is also, while there is an ethnic component and racial component, there's also a social economic component as well. Teasing this apart, one of the things that we looked at were new cases for patients who identify as white, Asian, and Hispanic Latinx, new cases above the median income, and new cases from patients who live below the median income. And we can see that it's disproportionately affecting those in low-income neighborhoods who identify as Hispanic Latinx. Looking at other socioeconomic trends, we have observed that COVID-19 patients are increasingly coming from neighborhoods that are denser, those of lower education levels, those who have an inability to work from home, and those who have decreased levels of health coverage. And unfortunately, this has really created a perfect storm where it's disproportionately affecting vulnerable, potentially frontline workers who may have lower levels of health insurance. And currently, one of the things that we are exploring with the initiative is how can we identify these vulnerable populations, and also what are possible interventions. Talking quickly to make sure we have time for the fireside chat. Next, I'm going to discuss how our initiative was trying to leverage all of this data to identify those who may require a higher level of care or critical care. With the rapid spread of COVID, especially in China and Italy, both countries quickly provided early characteristics of their patients who were refractory to therapy. However, we quickly observed that there were two key challenges. First, there was almost too much heterogeneous data. We looked at all the papers in February and March coming from China and Italy and their early experiences, and we found over 70 clinical, laboratory, and imaging parameters that may be useful in separating these groups. And as you can imagine, for a clinician, realistically digesting and integrating all this new data in real time would be difficult, especially when you factor in a busy practice. Second, and I think this kind of talks about what Dr. Geschwind talked about, is our populations are very different. Compared to early data from China and Italy, our patients tend to be younger. Our average admitted age is sub 50. Also, they have different comorbidities. If you look at the early data coming from Wuhan, the clinical comorbidities that they found significant were hepatitis B and anorexia. For the UCI cohort, we had very few hepatitis B patients. And in fact, we were looking at obesity to be a predictor, which is the exact opposite. And lastly, we know that based on the earlier slide, we have a very heterogeneous demographics. It's comprised of multiple ethnic groups from varying different social economic backgrounds. And so to meet this challenge, our team quickly trained, validated, and deployed a risk calculator, and the link is live here. Using EHR data, we mined and identified several clinical and laboratory predictors. And so what we did was we trained across our initial UCI experience. We had an internal validation, and then we also had an external validation provided from Emory. And we can see these were the initial results between our training set, our validation set, and our external validation set. The results held up relatively strong. In terms of the inputs, so things that we found to be significant were age, the number of comorbidities, including hypertension, diabetes, obesity, asthma, etc. BMI, clinically, those who presented with a high respiratory rate, routine lab values that may be seen in sepsis, and also labs from the COVID severity screening lab panel, which are markers for inflammation. And so this has been in use. I occasionally get text messages from clinicians who are using it. And the goal of this is really, can we have an early warning system, within a few days window that, these are patients that may be downtrending, maybe need to be more observed, and can this help augment, and can we capture more and do a better job of identifying patients who may be vulnerable to just supportive care. And so in summary, this is really a brief discussion about how precision health with AI can be quickly mobilized to improve health and well-being for our COVID-19 patients. And I think I'll let that lead into the fireside chat. Peter.
All right, thank you very much, Dan, and actually all of our speakers. You know, we're getting right near to the conclusion of this session, but I do think there are some interesting discussions and threads that have popped up. And so we will probably go over just by 5 or 10 minutes to ensure that we have an opportunity to engage with our esteemed guests here. So I'll start off first here. Again, there are many questions, so I'm going to try to do my best to sort of summarize the main points and the gist. But I think one good initial opening question for both you, Dan, and Xiaohui, actually. So we're hearing a lot, right, in the first talk about precision health and the opportunities in that landscape. And obviously, Xiaohui did a great job of highlighting the potential of machine learning and deep learning specifically. Now, I would ask you both the same question. What do you think is sort of unique on one hand about precision medicine? What is it that machine learning or deep learning can bring to the table that perhaps was lacking in prior generations of analysis? And conversely, maybe for you, Xiaohui, what makes machine learning different? What makes precision health such a great target for applying some of these tools? I mean, you can really apply the technology to anything under the sun, but again, precision medicine just happens to be a great fit. So, you know, either one of you guys, one of you can take a stab here, and then we can move on to the other.
All right, so Dan, I'll get started, and then maybe you can comment on that. So, I think here,
Geschwind, right? There's 2 Dans here.
Yes, and there's multiple Dans here, so.
Yeah.
All right, so for Peter, your questions here, I think the reason why people are excited these days is because there are a number of reasons. First of all, people do make a lot of progress in terms of the model, in terms of the algorithm that we made recently. And previously, in the old days, maybe even 5 or 6, 7 years ago, we have those traditional models. And those traditional models in general, doesn't handle the large data sets very well. And then nowadays, we have this deep learning based model. There are particular advantages that they handle a large amount of data sets. And that would be very useful for application in medicine, even in biology side, because over the past few years, and people generate a huge amount of data sets, and that's a huge interest in terms of looking into the data sets here, try to identify kind of useful information from them.
Dan Geschwind, you're muted.
Dan Geschwind.
Oh, I didn't think that was. I thought it was a Daniel Chow question because he's a, computer guy. But I would say that these deep learning approaches, not the kind of standard random forest, support vector machines, those kind of things. These deep learning methods, for reasons that maybe some of the mathematicians among you can explain, seem to perform much better with these big data sets than we can do on our own. And so they're picking up probably latent factors that we're not, and kind of almost inferring them, I guess is my way of, thinking about it, things that we're not measuring or paying attention to, and finding connections that we're not. The danger with them is that if the samples that you use have, some of those latent things may be, for example, I'll give you an example. Let's say you throw all the data in and you want to know the difference between schizophrenia and controls. But you haven't paid attention to the fact that, well, the schizophrenics are getting a medicine that the controls aren't getting. And so when you actually put everything together, genotype, transcriptome, metabolite, all that, part of it is being driven by the medicine, right? If you know that, you can remove that. But again, machine learning depends really exquisitely on the balancing of groups and things that are being compared. And so that is a potential pitfall with some of the methods. But again, that the people who are sophisticated understand, but that people who aren't sophisticated, who are outside the field don't. And so I think one just has to be, careful with it. But to me, it looks extraordinarily powerful in that, and it's predicted, we've seen things that I just would never have predicted. For Ron Halperin has used it to, another example would be post-operative kidney failure or heart attacks. People dying on the operating table has changed in the last 30 years, like from when I was in medical school. Like it went from like, I don't know, I'm just going to make this number up, but it was around 1%. And now it's like 1 in 10,000, it's less than 1 in 1,000. It's gone down 10 to 100-fold. It's been almost obliterated, people dying on the operating table. But post-operative complications have not really changed at all. That hasn't been budged. And so one of the things that a lot of people are working on in anesthesia is how to predict. And then if we can predict, can we intervene? And so people have used this now. There's some folks at UCLA and other places to actually identify really, really high-risk groups that can predict and the key is apply it to a new group and it can predict that group. Now the question is, let's say you can predict it. If you knew that, do you have an intervention and a bunch of things that you can do to change their outcome? And that's what we're actually testing right now. We're actually doing a kind of implementation trial to see if we can implement something to change that. But that's a big, so there are multiple steps. And I think it's essential that, just like you showed, and it was very, very elegant, that the machine learning people who are thinking about this are embedded and working closely with all the clinicians, either are clinicians or are so embedded that they don't make, novice mistakes that might get made, and also tackle problems where you can predict something and there actually might be an intervention for it. Like if we predict an asteroid is going to hit next year, we just might not have an intervention for that. So why we want to know? So anyway, I hate to, I don't know.
Yeah, absolutely. I think that is a very comprehensive summary of the key points. In fact, one of the commenters here has a very similar observation at the outset of your answer, which is that the complexity of the data in part is one of the unique features about machine learning and deep learning. Perhaps the possibility of integrating data across different domains, across imaging, genomics, and other things like that. So I do think that seems to be a great and promising opportunity, and one of the unique things that machine learning and deep learning specifically can bring to the table. So, and again, for the sake of time, there's a lot of great questions, but I think one theme that did come up a few times here is this idea of collaboration, right? In order to do this work effectively, you do need domain expertise across multiple disciplines and again, a very collaborative approach to these problems. And so again, to both of you, what do you think the sort of next steps or kind of key collaborations that need to be built in order to drive this field forward? And specifically within UC system, is there something, somewhat of an opportunity perhaps to band together and synergize between what each group has to offer?
Yeah, if you don't mind, is it okay if I answer that? Yeah,
absolutely.
Actually, I think it's Dan Mercola, another Dan. Dans are important, this is showing. Has a, so yes, there actually are efforts that are being driven at the investigator level and at the kind of, I would say, let's call it, those who are leading various efforts, but not at the chancellor level. But like Dan Mercola has been working with Olivier Harmonsier at UCSD, and they have one of these UCOP small pilot grants I've been working on to get kind of genomic data together on cancer, and that's going to expand into a larger thing. So UC Health has been pushing a UC cancer-wide thing because we would have more patients UC-wide and do much better than MD Anderson or any of these other, or Sloan Kettering. And we're in another league when you put us all together. In fact, in funding, we are enormous. I forget what it is. Let's just say, I'm going to say I think we're 25% of cancer center funding in California at the UCs, but not of national funding, something like, it's some big number nationally. But within California, the UCs only capture 12% of the cancer patients. So the opportunities in these areas are extraordinary. And I think it takes both bottom up, like Dan Mercola's and our kind of efforts to connect, as well as top down. So and I think the top down becomes more difficult because being a public institution, UCs are generally, let's say, not as willing to take huge strategic risks. And it's more about managing things than strategic planning. And that's natural with large public institutions that have a lot of different constituents and things pulling. And so I think some of this is really dependent upon us. There is a UC-wide informatics, health system informatics that Atul Butte leads. And I know folks at Irvine and UCLA are kind of involved in that. I would say, unfortunately, that's not yet available UC-wide. It could be used by all of you guys, who just talked, right? To begin looking at UC-wide data and stuff, and many of us, and we just can't access it. We're not sure why, so we work on our own internal data in the moment, and UC and UK Biobank and other things like that. But what we have UC-wide is kind of like a UK Biobank. So some of that stuff is being built, and so I think if we lobby to get access to that, there is a de-identified data mart that's sitting there connecting all the UCs. We've started to work, Leslie reached out to me on the COVID stuff to have a UC-wide kind of COVID consortium, and they're different, in host genomics. And so we're pulling that together. The UC health system has, the leadership asked, what should we be doing in genetics and genomics? So Paul Boutros and I have pulled together folks from the various campuses to begin to talk about this. Again, it got derailed by COVID and also the fact that there's a cost to developing a UC-wide strategy, but there are discussions around this. And I think that if I guess the message would be, if individuals have specific ideas about areas that they want to pull together, that they can see where this would really help, they can talk with their counterparts at the other institutions and begin to pull together consortia. And I think those will be incredibly powerful, and also talk with the leadership like Leslie and Suzanne and other people at your place who can help you figure out politically how to navigate that. But I do think that our opportunities are truly extraordinary. So the health records have been brought together in a de-identified data mart. It's not the complete health record. And someday soon that will be made available to UC investigators. And that will be extraordinary, right? They'll be unprecedented. That'll be 12 million medical records and all that, right? And so to build on top of that, we need, UCLA, as the School of Medicine, made an investment in agreed that we should genotype our patients and have a biobank. Well, that's something that maybe every school can't do, but it's not that expensive. And it would hugely increase the value of all the other precision health activities going on. So again, I would recommend doing that because it's now about $40 a patient. Of course, you got to set up the biobanking and everything else. But again, we're happy. We have Arash Naeem has been working with UCSF to implement our consent process there, and we're really happy to do that. So I think there's a lot of momentum at kind of our level, the people who are on this, to kind of connect things and do things together. I don't know, again, that's not a great, I see working together is just kind of keeping, things moving ahead and not necessarily depending on, UCOP to give us opportunities, but for us to kind of create them. On the other hand, UCOP has done, UC Health with the genetics and with the cancer is moving to kind of connect the health campuses together, which I think is a huge move forward and will be great. Sorry for
no, there's a lot to unpack
that was
there.
Great. Thanks.
I'll just briefly share my own experience. I'm a machine learning, deep learning person, and I work primarily in the imaging space in radiology. And I know that between the radiology departments, as well as the pathology departments or the imaging-based specialties, we actually do have a pretty strong grassroots type movement, much like you're describing, between certain PIs or KOLs or different individuals in each campus wanting to come together one after another. I agree that the traction from a top-down level from UCOP has also not been, has not always been there. They're supportive and they're willing to help, but there hasn't been some sort of consolidated effort to really bridge all of our efforts. One of the things that sort of sprang to my mind as you were talking is that if there are these grassroots movements within each specialty, well, maybe now the next thing to do is to start to cross some of those specialties, right? Imaging and genomics, for example, that would be a very nice sort of cross-disciplinary type of initial foray into some of this work. And again, we have the imaging data ourselves on our side. So it's just now about marrying that together with some of that genomics and precision health work that you guys are doing. Xiaohui, I apologize. I don't know if you had something you wanted to add to that.
Well, I do have a lot of thoughts regarding that because I'm mostly in the computer science. So we are sitting in the other side of the campus. So for essentially based on my understanding, our students, they know this computers science very well. They know models well, and however, they don't have enough knowledge on the specific domains, right? In terms either in medical imaging or genomics or genetics. I think maybe medical imaging is closer to what their knowledge. This genomics and genetics is very far away from what their knowledge base. It's kind of in the past has been challenging in terms of how to engage those student training in computer science to work on this type of problem, because it's kind of hard for them to understand them, and then understand the problem. Because in many cases here, in terms of applying this AI to solve this type of problem, and in my experience, most of the challenging part is actually to identify the right problem to work on, and then once you identify the problem, and then you put together a data to say if you can address that problem. And this initial stage is, in fact, is very important part, probably more important than later model building part of that. So however, that requires the domain knowledge of that. So it's certainly very challenging in that aspect. So I haven't seen many work, in particular, how to combine imaging with the genomics, right? So that part of this type of domains work in is not many out there. So then in terms of how do we improve this, I think what we are doing, I think those are precision medicine initiative or other kind of a workshop, other programs is very good in that aspect, bring people from different domains together. And at least they start with the discussion, and then maybe they can work on some problem together once they find common interest in that. But starting this have engagement, that's a very good thing, especially for people like me from computer science part of that, because otherwise I was not aware of these problems. But many of these problems are exciting, so this kind of engagement is very, 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 very helpful in that aspect.
Could I add?
Dan?
Yeah, I was just going to add something to that too. For example, in this imaging area, that's a perfect area for the kind of cross-UC collaborations, because there's this enormous challenge of trying to get these data in a quantitative form where they could be broadly used. Imaging data right now is kind of like, one could say that you don't want a novice jumping into functional genomic data and just trying to use it, because that has all kinds of problems. But I think imaging data, it is another magnitude of problem of kind of making it interoperable and usable kind of cross-system and all of that. And so the kinds of things that we could do to kind of make all of our images, on the same platform. And let's say somebody like me wanted to do something with them, it's kind of impossible. I have to talk with Alex Bowie, who's, your equivalent at my, institution, or, find other people. I guess for the moment, it does require real experts involved in these multidisciplinary collaborations. But I think one of the challenges is to get the imaging kind of like the clinical phenotypes, maybe to a point where there are aspects of it that are extracted. Is that possible? Do you guys think that that's possible, or you think that's?
Yeah, well, so that's actually my personal interest is doing just that segment of it. I'm a physician, and I'm also a deep learning guy, right? That's a very unusual place to exist, and because of that, I find that that's where I should be living. That's my role, is to sit on the hospital side of things, understand how to extract data in a way that's organized, that's objective, and to actually slice a problem such that my colleagues in computer science know exactly what needs to be done, right? If someone says, I want to build a tool for stroke risk prediction on CT exams, right? That's so open-ended. That's very, very hard for a non-clinician to understand. But if I tell Xiaohui, well, I need a tool for segmentation of collateral vasculature on CTA. It's going to be a segmentation tool, a UNet decoder architecture, and I can give you these masks in this type of format. Well, then Xiaohui doesn't need to actually know the specifics about how collateral vasculature affects recovery and stroke, but he knows how to solve that problem I just posed to him, right? And so I do think that translation, that sort of ability to go from all the data we have in the medical records, which is really just sitting there, to now in the hands of our experienced developers to build algorithms, right? That translation is oftentimes a big bottleneck. So absolutely, I think that's one of the primary focuses, in fact, of the work that we're trying to do.
And then integrate that with genomics and, you know, some of these functional genomics that we've been talking about would be
Absolutely
amazing, right?
Yeah, once the quantitative information is extracted from the imaging in a way that is, you know, what's the word? I can't think of it. You know, kind of reliable and the same across the, you know, then the genetics becomes pretty, you know, there are these consortia like Enigma and others where they've, you know, been able to do that. And of course, we'll have clinically relevant phenotypes, which make it even more interesting, I think.
Yeah. So I think maybe this is a good point to stop and thank very much our speakers, and thank you, Dan Geschwind, for being our first distinguished speaker here for this series.
Distinguished means gray hair.
No.
That's what I realized. So
Nope, it's the track record. Anyway.
No.
But thank you again, and appreciate everybody joining us, and thank you to the speakers. And
thank you.
Yeah,
thanks
thank you.
again.
Super
Take
interesting.
care,
Look forward
everybody.
to more interactions. Hopefully this is the
Definitely.
beginning of more
Yep.
cross UC. Thanks.
Yep.
See you guys.